This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 230, recorded on April 26th, 2013. Hello, everyone. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Hi there, Vincent. How are you?
Good morning, Professor. How are the poxes?
Oh, the poxes are just grand. We've been having a great time.
Can't imagine when you retire and you don't have poxes anymore. What are you going to do?
I got other things I can do. I'll be okay.
You're at peace, I can tell.
I am at peace. Everything is just going to be, Susan tells me everything is going to be just fine.
Yeah, that's good. That's the way.
And she's a mom, so she knows.
Yeah, right. That's what you tell kids a lot.
Right.
And they look at you askance. You can say that because you're old. Things were not the same. Okay, also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
Hey there.
It is 32 degrees Fahrenheit, 0 degrees Celsius.
What's going on out there?
Oh.
It's bright and sunny. It's going to warm up to
Why is it winter? That's terrible. I'm so sorry.
We got 70 degrees, clear blue, sunny skies. So that's amazing. That's like almost a 40-degree difference.
Yeah.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
What do you got out there?
54 Fahrenheit, 12 Celsius. It's not bad at all. Clear, sunny.
Car 54, where are you? You guys
That's right.
are too young to remember that.
I saw it on reruns.
Oh, wait. My computer just updated. It's now 42 degrees.
All right.
That's a big
Celsius.
difference.
That is a big difference. Yeah.
Oh, wow. But it's still chilly.
You feel better?
Oh, yeah.
We are at
Good.
14C here, Celsius. Clear blue skies. Very nice day today. If it weren't for the traffic, it would be perfect.
I decided after the last episode that TWiV is the weather in virology.
Yes.
The weather in virology. Very good,
Yes.
Rich.
Yeah, every now and then.
You got Alan rubbing off on you. That's cool.
God.
All right. We have some news. Who put the Francois Jacob article there?
Me.
I figured. You want to tell us about it?
Well, Francois Jacob died last week at the age of 92, I think. There's a nice little write-up by Carl Zimmer that I posted the link to that shows up in a National Geographic blog, believe it or not.
It's interesting.
And it's a nice little obit, I guess. Francois Jacob, he and Jacob, I'm blanking on Jacob's first name.
Francois Jacob and something Monod.
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 Jacques Monod?
Anyway, Jacques Monod.
I think so.
I think that's right. At any rate, they are
Monod.
famous for figuring out what an operon is and how it works. And their work was focused on the beta-galactosidase operon. That's, not necessarily that particular detail is not necessarily all that relevant, but basically that's an operon that in response to environmental conditions, that is whether or not there's glucose around as a carbon source or whether or not there's lactose around as a carbon source, it regulates the synthesis of an enzyme that is necessary to metabolize lactose. And they figured out using genetics how this thing turns off and on. And so the fundamental principles of a repressor molecule, that is a protein that binds to DNA and shuts off transcription of a bunch of genes downstream, were developed by these guys. It was the first example. And, I've been thinking about this since this came out. Basically, all of the transcriptional regulation that goes on is just variations on this theme. This was a really incredibly fundamental discovery. And this obituary is cool because it tells a story of him telling his wife about how he thinks he just discovered something, and she said, "Oh, haven't you told me that before?" And the bottom line being, it's so elegantly simple that it just seems obvious. Like, I must have heard that before.
Yeah.
Right.
Terrific.
This one's in the textbooks.
So he was quite a scientist and had an interesting life that's highlighted in this obituary.
I like this part here. He squirmed in a Paris movie theater. I thought you only squirmed in New York movie theaters. So this loom by Carl Zimmer, I thought it used to be at Discover magazine.
It was most, if not all, of the popular blogs from Discover transferred over to National Geographic. Discover has been, as they would say in the industry, reorganizing a bit.
Oh. Did National Geo buy them or something?
No. Discover got sold to Kalmbach Publishers. They're best known for Model Railroading magazine, I think. Yeah. They moved the Discover offices out to Waukesha, and then they chucked their bloggers over the side for the most part. I think the magazine's having some problems.
This is the kind of stuff writers know about.
Yeah, that's weird. I figured Alan would know this.
Yeah, I keep track of these sorts of things.
You're probably a member of the union or something.
No.
You don't have a card?
Don't quite have that. There's kind of a union for science writers, is the National Association for Science Writers.
Yeah, I was looking at that the other day.
Yeah.
I was thinking of joining, but I'm not a science writer, so I didn't.
I keep thinking of joining, too, and not getting around to it.
Around to it? I have one of those.
Yeah, I was given one a while ago.
Okay, follow up from Tim. Hey, TWiV team, just finishing listening to TWiV 229, although I've been a TWiV and TWiV listener for a while now. This is the first time I've listened to any of the Twix podcasts in the week it was actually recorded. While listening to Rich Condit's comments on how those involved in monitoring the H7N9 virus are doing a good job walking a fine line between unnecessarily overhyping and downplaying the risk of a pandemic, the name Bernardo de Bernardinis came immediately to mind. He and 7 other scientists were charged with manslaughter, not because they failed to predict an earthquake that killed over 300 people, but because they downplayed the probability of a major earthquake around the time that L'Aquila was hit, and were therefore found liable for the deaths because they had unduly reassured the public. Those charged with the job of predicting the next flu pandemic must be acutely aware of this incident, and as a result, I don't blame them for being noncommittal in anything that they say. Can any good come to a scientist who makes definitive predictions to the general public? If a more nuanced prediction was made, all subtlety would likely be lost to the ether that is mainstream science reporting. This is one of the many reasons the world needs TWiV. As I've mentioned in letters to both TWIP and TWIM, which are lost in the queue somewhere, I imagine, thanks for all of your time and effort dedicated to bringing your scientific insights to the rest of us. I think H7N9 is going to be a big problem, and you better get ready.
Yes, absolutely. As we've been saying.
That's a good point. I didn't know this story. Wow.
Oh yeah, that was a huge case in Italy that I think these guys are currently behind bars. I don't know if they've exhausted their appeals yet, but it's the details of the case are a little bit murky to me. There was kind of a big campaign of this is a horrible miscarriage of justice, but I gather that there were actually some arguments in favor of these folks having really downplayed the risk. So it's a little bit unclear to me.
So here on TWiV, we say we don't think the virus is going to transmit.
Right.
We don't say it's not going to. We just say, I say anyway, I don't think, but we're not making predictions, I guess.
Right, and I think we've done enough to undermine our own prediction.
Yeah. Okay, thank you, Tim. Alan, can you read the next one?
Sure. Catherine writes, Hello, TWIVers. I have a question about HeLa cell lines. Recently, a reference genome was published. While I think it should not have been done without the consultation of the family, that boat has left the dock. Vince's story about his 2 batches of HeLa cells, one making nice flat monolayers, the other not, made me think. If two lines behave so differently, how can anyone claim to publish a reference genome? What is to stop Vince, after talking to the family, of course, from taking his batch of HeLa cells, sequencing it, and saying, here, a reference genome for HeLa cells? I fail to see how cells that are grown indefinitely don't pick up random mutations, especially when those cells initially came from cancerous cells. To make my point more clear, maybe, how can a constantly evolving cell line that is in use in tens or hundreds of labs, all with different properties, have a reference genome? Thanks for reading the ideas that come to me at night. Catherine is in Korea, where it is rainy and 52 Fahrenheit.
All correct.
Yes.
Well, a reference is just that. Then everyone says, hey, I've got a line and it's got these changes compared to the reference.
Right.
It doesn't mean that it's Henrietta Lax's sequence. It's just something to compare to, right?
Right. It's like when you normalize your values in a paper, the values were normalized to such and such, but that's just the reference you picked.
Yeah. So
we had a presentation in Journal Club recently that talked about the karyotyping of a line or a population of HeLa cells. And it was like 86. Okay. So, I mean, even the ploidy is all screwed up. They got that particular lineage had like 4 of some chromosomes and, translocations and all kinds of stuff. And I'll bet you other lineages are equally screwed up in other directions. Yes. By the way, Vincent, we plated out our HeLas for you.
Yeah.
And they stick to the dish, and they're nice and plump and round.
They're round, huh?
They don't go flat at all.
Oh, man, it's just like the new ones we got.
And you can get them off the dish, but it doesn't even require trypsin. Couple of washes with what we call Versine, right, which is just PBS with some EDTA in it, and you can squirt them off the dish, and they will stick, but not hard, and they'll divide and that kind of stuff. But they're nice and plump and round. They don't flatten out at all.
Okay, I don't want them.
Okay.
Because when our virus infects a flat cell, it makes it round and plump, and that's how we can tell.
Right.
And
absolutely.
if you infect these round and plump cells, we can't tell that they're infected.
Right.
So anyone out there have a flat line of HeLa, let me know. I'm trying to find it. We had it for 30 years, and we lost it, and we need to get it back.
I'm sure there's plenty of flat lines out there.
You want it to be able to come from spinners, though,
yeah,
right?
And flatten out.
Yeah. Okay, well, I can check ours out.
You grow HeLas?
Not routinely, but I think I have some from Arnie's lab. They may have come from the same MIT set originally.
Okay. See, there's this perception in a lot of the public discussions about science that this is all rigorously controlled, and you would use everybody using the same cell line as using identical cells. But now you see from this discussion that it's do you have some of these? I got some of these. I've been culturing these for 30 years.
It's okay though.
And ATCC makes a case for if you want a certain cell line, you should get it from them. So when we get our L929 cells, we get those from ATCC. But spinners are something that, maybe they're in the ATCC collection. I guess that's where you got yours, Vincent,
yeah,
the ones that are the wrong ones.
Right,
yeah.
They have spinners. They call them S3, which is a clone that Philip Marcus made many years ago. And then they have non-spinner. They have non-S3 variations of HeLa too. And we've tried both. And the non-spinner don't grow well in spinner, as you would guess. And the S3 grows really well, but this version is round, and ours was not. So,
we have the same thing with, if we get the mouse 3T6 cells from ATCC, mouse adeno won't plaque in those, but we have some that we got from somebody else, and they plaque fine.
So one of the problems with these cells that we have is that they don't grow rhinoviruses very well, and that's a problem for us because we do half of our stuff on rhino, so we have to get the old ones back, and we need to grow them in spinner because we use so many. We need a liter of spinner a week and so forth. Nevertheless, Alan, we do get results and many people get results that are interesting despite these.
Of course, yeah, because what you do is you look at your results through that lens. You say, well, this is what happens in these cells, and a lot of the experiments that you're doing, it doesn't really matter terribly much whether it's this genetic background or that genetic background of HeLas, because you're growing up the virus. You're really studying its biology.
Right. But Catherine is right. These cells are full of mutations.
Yes.
constantly growing cells accumulate them, and that's when that's why when you initially transform a cell, you make it grow forever. It accumulates mutations and eventually will become cancerous because you need mutations in certain genes to do that. And they continue to accumulate mutations as we grow them. So the reference is as saying just randomly, this is the sequence and compare yours to it.
Yeah.
Kathy, would you like to read your follow-up email?
Sure. So I think this is about the fourth or maybe the fifth follow-up on the story of Welkin Johnson's influenza vaccine going into the eye and what could happen from that. And so you guys were talking about that and whether there's something in tears that's antiviral. And there is some literature about lactoferrin, which is in tears and a lot of other mucosa and most body fluids, that it's antiviral and evidently against rotavirus, enterovirus, and adenoviruses. And so I found a publication that reviewed it. But what had triggered me to think about this is that some Swedish groups, including Nicholas Arnberg in Umeå, had shown that some adenovirus serotypes co-opt this lactoferrin and use it as a bridge to enter the cell.
Interesting.
But if the lactoferrin concentration is really high, it seems that the excess lactoferrin reduces binding of the virus to the cell, and that maybe the lactoferrin binds to either the virions or the cells, but not both. And so at high concentrations, the bridging doesn't occur. And that for other serotypes, this bridging also may not occur. And so that lactoferrin is in such high concentrations in tears that it's protective in the eyes, but in other places like saliva where the concentration is low, it might not be protective. So
how interesting.
Yeah.
That's not surprising that there would be something in there. There's probably other things as well that are part of the intrinsic defenses, right?
Right.
So when infects your eye, does it remain localized or does it spread elsewhere?
Boy, you're asking me a question I don't know the answer to. But if you're talking about the ocular serotypes, my suspicion is that it stays localized. But I don't really know.
Okay. I was wondering if it went down the nasolacrimal duct.
Now that we know about that duct, we're always onto that. Yeah.
Well, the cool thing was last time is that this Fouchier had this published a paper last week where he talked about conjunctivitis and H7 human infections, and he thought that the duct would be a good. And so Welkin's original comment has huge legs. It's great.
Yeah.
They call it a long tail. Okay, I think we should just bring everyone up to date on H7N9. And if you have been living in a cave, that is a avian influenza virus that has infected so far 109 people in China, known cases, 23 deaths. And WHO every week puts out a cool summary of what's going on. They have 3 slides in each file. The geographical location, which you can see is in provinces that are mainly on the east, or entirely on the east, except for, I guess, Beijing, which is a little bit in. And as Alan said last week, there's just not enough people maybe out west.
Yeah.
They have the number of cases in a table, and then they have the epidemiological curve, that is the cases according to date. It's kind of cool. And, of course, this just came out yesterday, the latest one, that's number 3, and it's already out of date.
Right.
But that's okay. So check that out. There was a group of very important people who went to China on an investigative mission, and there's a report on that in Science Insider. They said it's an unusually dangerous virus for humans because it's easily transmissible from poultry to humans than H5N1. And in that same article, there's a link to the CDC in Taiwan where they had an imported case this week, someone who had been in Shanghai, I believe, for a couple of days, and he went back to Taiwan. He was fine. Then a couple of days he got sick, and H7N9 was identified in him. And he had contact with something like 138 people in the course of his living there and then going in hospital and so forth. And some of these people apparently developed influenza-like illness, but I don't know what the outcome is there. Now, this virus supposedly doesn't transmit among humans, at least yet, so we'll see what happens. I look for
the thing I'm having trouble with was this notion that it transmits easily from poultry to humans. Is that it doesn't seem to be, really obvious in poultry, right?
Yeah.
Right. So the
What-
Beijing Dateline today from the Xinhua News Agency, 68,060 samples have been collected from poultry markets, habitats, farms, and slaughterhouses. 46 have tested positive for the virus. 44 were from live poultry markets. One was from a wild pigeon, and one was from a household farm of carrier pigeons.
Sorry, where were they collected? All over, or just in certain areas?
This news thing doesn't say. The other one from ProMed Mail was a couple days ago said 39 cases from 48,000 samples from more than 1,000 poultry markets, farms, and other sites. So, you might have 1,000 poultry markets just within a small area in China, I suppose. So I don't know how widespread they're testing.
Yeah,
but they're testing a lot.
It doesn't sound like it's coming from any of those sources to people, right?
Well, I suppose the poultry is the most logical source, but for me personally, the numbers don't add up.
But the sequences would suggest that that's the source, because a lot of these, at least the initial sequences from the pigeon and the environmental sample at the market in Shanghai, they were very similar to the human sequences.
Right. I think one of the important observations for tagging this as an unusually dangerous virus is that these human cases are cropping up at a faster rate than the H5N1 cases did. I mean, we have 109 cases in a much shorter period of time than the H5 cases showed up. So,
and it's not all poultry workers.
Right.
This appears to be, some of these people had little or no direct contact with live poultry, as far as we can tell. So there's this definitely appears to be easier to get from poultry to humans.
Yeah. Right. Makes the jump easier.
I have a correction. The Taiwan case, he was in Jiangsu, not Shanghai. And he could not recall having any contact with poultry, at least none that he will tell us. The Nancy Cox from the CDC said we can now understand that the likely source of infection is poultry. I guess that's based on the sequences.
Right.
But if you don't have contact with poultry and it's not going person to person, then how did we get it? So that's a mystery remaining to be solved.
And it's not in pigs, right?
Right. This Xinhua thing also says the virus has not been detected in pigs.
Can you put that link in the show notes, Kathy?
Yes, I can do that.
Thank you. We will put it in the show notes. And that's our week update. A little quiet this week, I guess, on the H7N9 front.
Yeah.
All right. I have a story which I thought would be interesting for us to comment on. This has nothing directly to do with viruses, but could in some way impact on it. And many people may know that the United States Supreme Court has heard arguments recently, April 15th, over whether human genes can be patented. And this comes from a case where Myriad Genetics was sued. They had patents on two genes, BRCA1 and 2, which are, when mutated, may cause breast cancer. And the plaintiffs said, we don't think this is right to clone genes. And so they went all the way up to the Supreme Court.
Mm-hmm.
So what do we think? Should we be cloning? Yeah. Should we be patenting human genes? And if by extension, should we patent viral genes? Of course, that's not being considered here. See, I'll give my opinion. I don't think you should patent human genes because you're not discovering anything. I think you could patent the use of the product for something, or modifying it for something, or purifying it in a certain way, a method. But just patenting the gene, I think not. But
I agree. Just patenting the gene doesn't make any sense to me.
Right. I agree.
Yeah, and legally, my understanding is for a patent, there has to be some kind of inventive step, and it can't be obvious. And obvious has a specific legal definition, but you can't just pick up an apple that fell from a tree and say, I want to patent this. It's a naturally occurring thing. You can't patent it. But what you can do is, if you are a plant breeder and you selectively breed a strain of apples and you put in an inventive step where you are testing the apples coming off the trees and you find one that's particularly sweet or particularly firm for making pies or whatever, and then you produce a new strain of apples through this inventive step, that was not obvious.
Yeah,
that wasn't something that was just sitting there in nature. You made it. You can patent that, and in fact, patenting plant strains is, I think, as old as the patent system.
Right.
That's an established thing that you can do, and that is the mechanism. My understanding is that that's the legal rationale behind things like the Harvard oncomouse, which was one of the first patents of this sort. I think there are some bacterial strains that were patented before that, but the legal reasoning there was, well, you're not patenting mice. You're patenting a specific strain of mice that you made through this inventive process, which required a heck of a lot of work. And it was not at all obvious that would exist or that should exist, and it was created for particular purposes, and so therefore that's patentable. What's at issue in this case, so just the basics here, the company that is patented, that's the plaintiff here, is a company called Myriad Genetics. They patented two genes called BRCA1 and BRCA2. These are associated with a certain rare but very severe familial breast cancer. And they patented, I believe they just patented the genes.
Mm-hmm. That's correct.
This is something, even this type of a debate is not new. There still is a company called Human Genome Sciences, but if you remember a few years ago when the genome sequencing efforts were underway, HGS collaborated with Tiger and with Celera, Craig Venter's two efforts. And HGS, I think initially the idea was that they were going to analyze some of Celera's gene sequences, find ones that would be commercially useful or important, and just selectively patent those. But in practice, what HCS ended up doing was basically taking Celera's output and plugging it into the patent office. They filed thousands upon thousands upon thousands of patents for genes that they had virtually no idea what they did. And I think this is one of the things that caused the split between those two companies, although I'm not deep into the history on them. And I don't know, maybe we'll get an angry email from Craig Venter correcting us. But the upshot of that, as I recall, was that there were some rulings and there was some backing off of what could and couldn't be patented. But as this case illustrates, that issue was never completely legally settled. So it's kind of been in this gray zone, and the patent office is, I believe, sitting on a lot more patent applications besides just this. And Myriad, of course, in patenting these genes, wants to be able to continue selling their test for them.
Yeah, that's the key. I mean, you can patent the test. I don't see why not, but why do you have to have a patent on the gene? Right?
Exactly. I think the test is the important part here.
Yeah, of course. For the test, there's going to be prior art, so that's more complicated.
Of course,
right?
Testing for a gene is not a new idea.
Yeah.
So I think this was supposed to be their unique intellectual property that would give them an edge.
Yeah.
And there are arguments on both sides of this about how either the ability to patent or the inability to patent will either promote or inhibit progress in research and science and stuff. And you could go through all of those arguments, but you can construct logically sounding arguments on both sides of that issue.
Yeah.
So we have several links in the show notes, and I particularly like the blog by Michael Wozniak, who, in fact, is on a couple of patents himself, but now he's made this about face, and just eloquently talks about it and quotes several other people. And one is from Michael Spector, who says, a woman who inherits a harmful version of either of the genes that Myriad has under patent, for example, is 5 times more likely to develop breast cancer than a woman who does not. She is also at significantly greater risk of developing ovarian cancer. Women who want to know whether they possess those harmful mutations have just one way of finding out, by taking a $3,000 blood test offered by Myriad Genetics. To seek a second opinion, their only option is to pay to take the test again. And then I can't remember quite where it was, but someone said, basically, when we get to the $1,000 genome, we're going to have way more information than this $3,000 test. And yet, when we get the $1,000 genome, at least the way I understand it, as it stands now, you wouldn't be able to release or deal with the results from the BRCA1, BRCA2 sequence.
Yeah, right.
So it's really a mess.
It is.
Yeah.
Wow.
But and here was the other one that Francis Collins says: if we end up with a $1,000 genome that comes with a $5,000 fee for royalties, that's not okay.
That's good,
right.
So that could be the other issue.
I mean, the $1,000 genome is problematic also because to analyze it, not everyone is going to just be able to analyze it. So there will be companies coming up to do it for you, and then they will charge you a lot of money, most likely.
And each company is going to have its own flavor of analysis that they're all going to claim as superior.
That's right.
But a lot of those, and a lot of those analyses will use proprietary approaches, I'm sure. But that's kind of a different ball of wax. The issue here is if somebody can own all rights to BRCA, then that's going to be a huge problem. If you have your genome sequence, why are you not entitled to know the sequence of those genes?
Exactly.
That seems fundamentally wrong, and that seems like a major slapdown of innovation.
Yeah, this is a good column by Wozniak. You should read it.
Yeah, I think this actually is probably the best summary of it that I've read.
Yeah. Apparently, the court is considering allowing cDNA to be patented, which doesn't occur naturally, so it could be patented. Which I think.
Yeah,
is
I think that
crazy.
Whether or not it occurs naturally is a problematic boundary that they're setting. Okay.
I don't see why cDNA would be patentable, right? I mean,
No.
for a gene, that doesn't make any sense to me.
Right.
I mean, that's like saying it's like the messenger RNA is not patentable, but the cDNA is. I don't think so.
It doesn't make any sense, but it seems like an artificial distinction, right?
Yeah. Well, they're doing what they can to draw this boundary between what exists naturally and what is man-made.
Yeah.
And if that means that cDNA is patentable, then I think they're going to have to rethink it.
This is typical of the sorts of really, really fine shades of distinction that the Supreme Court frequently has to weigh into. I mean, when they're doing their job.
Yeah.
It's usually a case where it didn't end up there for being easy to decide.
Yeah.
Right.
And really, the essence of this case is what is an inventive step? What's an invention?
Mm-hmm.
And as you just pointed out, Rich, if just the DNA is the cDNA patentable? And if the cDNA is patentable, then why isn't the DNA patentable? And you get back and forth in that.
I would say basically, well, I don't know if this will work, but if the sequence exists in nature, all right, then your ability to copy that sequence and make a permanent record of it in a cDNA is not patentable. Okay, but I don't know. Maybe I would head for Washington and argue.
And depending, your ability to copy it and sequence it could be patentable if you use a unique process to do that.
Right.
But patenting the process,
right?
You're not patenting the gene itself. And I would argue that cDNAs do exist naturally, just not as DNAs.
Whatever happened to the original
That sequence exists.
When cloning first came, I think there was an attempt to basically patent the cloning process, right?
Mm-hmm.
Yeah.
That is recombinant DNA technology. I don't know whatever happened to that.
Well, those are probably expired by now, right?
Those are definitely expired, but there were, in fact, a lot of patents, a lot of very valuable patents on what we now consider fundamental techniques. But in all of those cases, those were processes that were that were really innovative steps that
Right.
involved, people pipetting stuff and doing things that don't occur in nature.
Right.
In this case, Myriad is saying they own that DNA sequence,
Right.
which is a different sort of argument. The claim is not we have a special test for it. It's just we own the sequence, which I think is an error.
All right, so here's the question. What is one of the top grossing biotechnology patents of all time?
Restriction enzymes?
No.
That one was.
It's the one here at Columbia. This
Yeah, I
Silverstein and
know.
Axel.
Saul's and Richard's patent.
Co-transformation, which is long
Co-transformation.
expired, but that was a big one. Just in the interest of disclosure, I have a few patents for which I am very unhappy about, and one of them is on polio cDNA, and another one's on some other viral cDNA, which are expired and never made a penny for anyone. And I have I had a patent on the polio receptor transgenic mouse, which was also useless. And
no, that mouse was not useless, but the patent was worthless.
Yeah, because Europe doesn't recognize transgenic mouse patents, and so whoever wanted to go around it just went to Europe. And the only thing these patents did was to restrict people from using the technologies, and for which I don't really like. All right, I think you develop something, you let people use it, and I would never, ever, ever patent anything I did here at Columbia again. For the record, if I invented something, I wouldn't tell anyone. I'd publish it, and you could do what you want with it, because I'm so disenchanted with this patenting of stuff that, we have a patent office here, and they always say, "Oh, if you develop something, tell us about it." No way. I would never. And I've met the patent guys here, and they talk to me, and I say, based on my experience with you, I would never tell you anything again. I am under no obligation to tell you what I've developed. So there. I really think that when you do this stuff, you should let people develop. I don't care if they make a lot of money on it. Good. Help humanity.
I agree.
Did I tell you how I felt about this?
I think you got the best across. Yeah.
There is one downside to that, though. If you develop something that is genuinely useful or is potentially genuinely useful and you don't patent it, it becomes a lot less likely, if it's something that would have to be picked up by a company to be translated into a useful product.
Yeah, you're right.
Companies are a lot less willing to do that with stuff that's not patented. And in fact, that was the entire rationale of the Bayh-Dole Act, which set up this whole patent office at your university and other research universities, was to patent the stuff that was done with government funds, specifically to encourage companies to pick those things up and commercialize them because of this perception, well, actually this reality that there's this valley of death between basic research and translated research. And to get across that, one of the things that you need is some kind of property that you can transfer to the companies that they will be able to use.
Well, the thing is, Jonas Salk developed a polio vaccine. He didn't patent it, and 6 companies made polio vaccine and sold it.
Yeah.
So there, it didn't inhibit it. If you make something that's really needed, right? I mean, I understand your argument, but
right. But in Jonas Salk's day, it was possible for a basic researcher at a university or a nonprofit institution to do the entire pipeline and produce a finished product.
Yeah.
Salk's vaccine was not, I've found this interesting phenomenon that might be commercializable if you spend another $300 million on it. Salk's vaccine was, here's a shot in the arm that prevents people from getting polio.
Yeah, I got it. You're absolutely right. My position is untenable, but I'm just pissed off.
No, I understand. I totally understand where you're coming from.
And I'm not doing anything anymore anyway. I'm just doing podcasts, so what's the difference? I can say whatever I want.
Back in the BRCA 1 and BRCA 2, one of the commenters on one of the blogs points out that the patents on these begin expiring next year.
Wow.
Yes.
But whatever the Supreme Court decides on this is going to have implications beyond just these 2 genes, potentially, unless they make a very narrow decision.
Yeah, but if they allow you to patent cDNA, then it's an easy way around it, right?
Mm-hmm.
So they shouldn't do that.
Except that with the $1,000 genome, the clock is also ticking on this because very soon there won't be anything patentable left. I mean, the reason Myriad was able to patent BRCA1 and BRCA2 is that these gene sequences were not generally known when they first patented them.
Well, one of the things I read said that essentially the whole genome is patented now. I mean, little snippets here and there, and add it all up, and it's the whole thing. I can't remember. I can't find where that was.
That goes back to that Celera Human Genome Sciences era.
Yeah.
That a lot of this stuff has been patented. The issue is that most of those patents have never actually been tested. So people commonly misunderstand that if the patent office issues a patent, that becomes the law of the land. It does not. If the patent office issues a patent, that says that in the opinion of the patent office, you have invented something. It's not actually decided until somebody sues you, and it goes to court, and the courts end up deciding whether you've actually invented something. So all patents are really kind of probabilistic.
That's interesting.
They can end up in court and get overturned, and this happens commonly. I mean, the patent office obviously takes measures to try and, do their due diligence and make sure that this will be an enforceable patent. But ultimately, you don't know if the patent is enforceable until it's gone to trial. And a lot of them never come to trial. People just say, well, our lawyers think we'd probably lose if we went that route, so we'll just license the patent. But it depends on the situation.
That's what Apple does. They patent everything, and then if they get challenged, a lot of them are thrown away.
Yeah.
Speaking of $1,000 genomes, so I had my son's DNA sent to 23andMe for just a scan of known polymorphisms and just got the results back. It's pretty neat being able to look through and see what mutations you have or have not. And they give you a frequency of penetrance that you might expect. You have a 5% chance of getting Alzheimer's based on this sequence. It's kind of interesting to go through, because I can imagine having the whole genome. You could look at all your endogenous retroviruses.
Yeah.
So, all right, let's do some virology. Wait. Yes, let's do some virology. No, wait, sorry. We have a paper that just came out this week as a letter in Nature. It's called Receptor-Binding by a Ferret-Transmissible H5 Avian Influenza Virus. And the authors are Zhang, Coombs, Martin, Liu, Xiao, Macaulay, Loescher, Walker, Collins, Kawaoka, Skahel, and Gamblin.
And it's from the MRC labs in Cambridge, mostly.
And then, of course,
I'm sorry, not Cambridge, Mill Hill.
Mill Hill. I haven't seen John Scahel's name in a while. Wow. Some people in Switzerland, Novartis, and Madison, of course, Kawaoka Lab, and China. So this is H5N1 back here, and this is a study.
Yeah, I remember H5N1.
Yeah, remember that? It's kind of getting overshadowed, isn't it?
Yeah.
This is a study on one of the viruses that was selected by the Kawaoka lab in that one of the two controversial studies that we talked a lot about last year. It was selected. It was an H5N1. Actually, it was an H5 on a background, I believe, of another virus, right?
Yes. It was on the background of a
H1N1.
Something like that. A background of a virus that I believe was already transmissible in ferrets.
Yeah. So they put the H5 on and selected for viruses that could transmit via aerosol.
If you just stuck H5 in, that virus was no longer transmissible, but then you could select for mutants that would be transmissible, and so ask for the genetic determinants of transmissibility with specific focus on the HA itself.
Exactly. And this virus turned out to have 3 amino acid changes in or near the receptor binding site, and then a fourth one pretty far from it near the virus membrane, which was involved in fusion. We talked about that quite a bit.
Yeah.
And the issue here is receptor recognition. These influenza viruses, of course, the HA binds to sialic acid containing receptors, and the avian influenza viruses preferentially bind to alpha-2,3 linked sialic acids. And the human strains preferentially bind to alpha-2,6 linked sialic acids. And the idea is that in order for an avian virus to become a pandemic strain, it would have to be able to recognize alpha-2,6 linked sialic acids. So the aerosol transmissible H5 in ferrets, one of the mutations allows the switch in receptor specificity. It's a glutamine at position 226 of the HA. It's a change to leucine. And then there's some other changes as well. And it's useful to point out that this change, glutamine 226 Lu or N226L, depending on your preference, has
glutamine would be Q, wouldn't it?
QG, yeah. Polio cleavage site, QG, glutamine glycine. Q226L is found in other human strains that have believed to have come from birds. The 1957 H2HA, it has the Q226L, and the 1968 H3. And both of those hemagglutinins are believed to have originated from avian influenza viruses. So in this paper, they're looking at how this transmissible H5 binds sialic acids, and they look at the structure of sialic acids bound to the H5. It's pretty interesting stuff.
So the contribution here is that we're actually getting a quantitative picture of exactly how these mutations affect the binding affinities and et cetera. And it's they have a couple of different techniques here.
Go for it, man.
Which Rich will now describe in detail.
Right. Okay. Well, so
you got an hour, okay?
Right. So actually, what's important, what I get out of this, what's important to me is one of their techniques is used to assay soluble receptor.
Right.
That is the trimer.
Right.
The technique they use for that is called microscale thermophoresis, which is magic.
Yes.
And then they have another technique that they use to assay the receptor in the context of virus. Mm-hmm. So they build viruses that contain these receptors, and they can assay for binding affinities of virus to receptor-like substrates, and that technique is called biolayer interferometry, which is also magic.
Yes.
And there are Wikipedia links to these that I read with diligence. And, actually, the important thing here is that I believe that, I can't prove this, but I believe that a normal human being, and I regard myself as a fairly normal human being, given the time, could understand this.
Yes,
right?
Sure.
All of this stuff is like riding a bike. It just takes time and interest, and you can figure it out. But I do not have the expertise right now to understand this. Then what they do...
so we could just say they used biophysical methods.
Yes, they
methods.
did biophysics.
Right.
And then what they do... Now, interestingly, they do on the soluble receptor, they assay the wild type H5. They assay a reference strain that's a vaccine strain X31. They assay the transmissible H5 and on all of the different substrates. When they get to the virus, they look at only the wild type and the reference strain, and my assumption is that because they, in order to do the avian transmissible mutant, they would have to build that virus, and that becomes a P3 experiment. Mm-hmm. Okay, mm-hmm. But by doing the experiment, the measurements on the quantitative measurements on the intact virus, with two things that they've done the quantitative measurements on the soluble receptor, they come up with an algorithm for predicting how the intact virus would behave for the transmissible guy. And on their third quantitative figure here, they show that, and they show that their algorithm works for the known guys. And so they make they use the algorithm to predict how the intact virus would work for the transmissible mutant. Yeah. And what's interesting to me is, just conceptually, is that these are fundamentally different measurements because basically the virus is multivalent. So a small change in receptor affinity for the soluble receptor translates into a much larger effect, one way or the other, on the virus because you have many receptors together that are many virus attachment proteins that are binding to receptors.
Right, exactly. And in these models, their predictions, they can test with some of the viruses that they can grow, without high containment, and they have good agreement between the prediction and the virus binding and what they actually see.
Right, and that's crucial when you're developing especially an algorithmic technique like this, where you're going to simulate everything in the computer. Be that they show these predictions of stuff they already know, and it all lines up very, very nicely, which gives a lot more confidence in the ultimate result.
I wanted to throw in that thing about, a normal human being could understand this stuff because this is kind of off the subject, but it's my sense that all too often people who are not scientists see science as some special thing that is can't be understood by normal people, and that's just not true. Okay,
right.
It's just a matter of spending time with it. That's all.
Right. So in cases where we say they did math or they did biophysics, and we joke a little bit about not quite understanding this, it's not so much that we're proud of not understanding it. It's that we didn't have the time to delve into this particular subspecialty in advance of the episode. But if I could just give a couple of quick tips. I did not dive deep on this to figure this stuff out, but I frequently find myself confronted with this kind of thing. I have to do a story on something, and I am not really well versed in the science behind it. This comes up a lot with biophysics stuff, in fact. Wikipedia is a nice place to start a lot of the time, but for these types of topics, I find there really are two common types of Wikipedia articles. You either get a really brief blurb that's often just copied from someplace else, and in fact, for the biolayer interferometry technique, that's what you have. Something that I think was copied from the website of the company that makes this machine. And then the microscale thermophoresis, you get kind of the other problematic Wikipedia entry, which is a highly specialized, detailed description with, in the second paragraph, they're giving you equations for calculating some coefficient, and it's just really, really inside, and it doesn't walk you through the background that you need. So what I usually do is kind of skim through those things, and then I try and find out who uses this equipment and who makes it. And in many cases, especially if it's commercially made equipment like both of these, there's one company that makes each of these. There are two different companies. One makes each of these machines. I'm pretty sure that if you go to the websites of those companies, they're going to have more approachable and more in-depth descriptions that you can dig into.
Makes sense because they got to sell the machine.
They got to sell the machine, and they have to sell it not only to researchers who are hardcore in the field, but to researchers who are in adjacent fields. And sometimes I'll go to a website and there'll be interactive videos and all sorts of really cool stuff that you can delve through. Other times they will at least just have a plain English description of how the technique works. So those are a couple of things you can do to try and get at find an entry into this sort of material.
I want to just say a few things about this binding stuff.
Yeah.
And so the affinity of an HA for its receptor, there's an affinity constant that describes that, which is a combination of how quickly the protein attaches and how quickly it dissociates. So the on rate and the off rate. Okay, and those two together make the affinity constant. But as we know, viruses have multiple interactions with receptors, not just one virus attachment protein with the receptor. And so you always have to take that into account, and that will give you the avidity of the virus for the cell surface. And what they did, which I thought was cool here, they came up with a number, which they call the multiplicity coefficient, that you apply to the affinity data, and you can sort of get an avidity prediction from that. And for flu on the cell surface, this coefficient is 5.5. They say it's no more than that. So that implies that there's not a whole lot of HAs interacting with sialic acids, maybe 5 to 6. I don't know if that's a direct conversion. But for polio, we think it's similar. There are probably just 5 receptor binding sites interacting with the cell surface. And so then, as Rich said, you can then calculate how, pretty small differences in binding affinities, like 15-fold, completely disrupt virus binding in the context of a multivalent interaction like that. This is pretty neat stuff. Anyway, the conclusions, which are really the most interesting part, the one that I find really cool is that this transmissible H5, ferret-transmissible H5, it only has a small increase in affinity for the human receptor. Now, again, the affinity is going to have a greater translation into actual binding because of this math that we've talked about. Yep. But the affinity for the avian receptor is way, way down, the alpha-2,3. And what I thought was cool was that they make this argument. First, mammalian upper tracts have a lot of soluble alpha-2,3 sialic acids floating around in the mucus. And these would be inhibitory for an avian strain. And so this would make sense in a ferret that you would select for this kind of mutation. Otherwise, the virus wouldn't be able to infect. Does that make sense?
Right. Yes. Right.
So it's a nice way. You don't have to be hugely better with the human alpha-2,3 sialic acid, but what's really important is to get rid of the alpha. Did I say alpha-2,3? I meant alpha-2,6. You have to be much worse at binding to the alpha-2,3. And the corollary of that is if this kind of virus arose in nature, it would have to be in a host that allowed alpha-2,6 interactions.
Right.
Couldn't happen in a bird, right?
Right.
Unless it was a bird that had a lot of... They give some examples at the end of some birds that have lots of alpha-2,6. Or was it a quail quails or mammals like pigs and humans. So that last step before getting into people would have to be another host. So that's pretty interesting, I thought.
Right. Now, is there also a downside to the virus for the compromised alpha-2,3 affinity in a human host? Because there are some alpha-2,3 receptors around. How does that impact the biology of the virus?
I don't know the answer because, the big assumption is that you need really good alpha-2,6.
Okay. And
down on alpha-2,3, some of the other studies that we have talked about where they have selected alpha-2,6 binding, there is a corresponding decrease in alpha-2,3. So it may be that structurally you can't do both, right?
Okay.
I don't know.
Yeah, and the implication there would be that the virus will be less suited to birds.
Yeah.
So it's it has to... if I can anthropomorphize a bit, it has to make a leap of faith in order to make it into humans and sustain in humans. It has to lose a lot of its ability to be in birds. So it actually has to make a jump.
Now that's interesting, actually, because it's got to do kind of everything at once.
Right. Right, and if it becomes transmissible in humans, then it can spread in humans, but if it's lost the ability to spread in birds, then it can't go back to its reservoir host.
Interesting.
If that's correct, which this paper certainly seems to imply that would be the way it would work.
The other cool thing, which I thought was worth mentioning, they did these affinity studies with a wild type H5, a Vietnam isolate, and this, as you would expect, has really strong avidity for the avian sialic acid alpha-2,3, but almost none for alpha-2,6. All right, so they said this is unusual among avian viruses because usually they are able to bind somewhat to alpha-2,6 because birds have some alpha-2,6. Right. They're not devoid of it, so that's kind of interesting. Then the rest of the papers are structures, three-dimensional structures of the HAs and sialic acid complexes. And these are pretty neat. So first they do transmissible mutant HA, then this Vietnam HA, they're both H5s, and a different H5, a turkey H5, together with human sialic acid analogs. Okay? Alpha-2,6 linked sialic acids. And these show that the sialic acid is fitting in the pocket in a very different arrangement compared with the way wild-type HA complexes with sialic acids. All right? So the transmissible HA binds the sialic acid in a similar way in the pocket as in human strains from 1918, 1957, 1968, 2009, and that's really different from the way that the non-transmissible H5 binds the human receptor. So the selection in ferrets has allowed this HA to bind sialic acid in a way that resembles the way human strains bind sialic acids, and which is very different from non-transmissible H5. Is that clear?
Mm-hmm.
It's a little,
I think so.
It's a little tough. Then they also looked at avian. Let's see, the loss of avian receptor binding by this transmissible mutant HA. So they determined the structure of that together with avian sialic acid, and it binds very differently from the wild type H5. It binds very differently to the alpha-2,3, and this is probably in part because of the change, this glutamine change at 2,2,6, and they have a discussion where they think maybe some of the other mutations might make a contribution. So that's pretty neat too. So one thing I thought was lacking here, they don't look at the effect of the individual HA mutations on either the affinity or the structure, the interaction with sialic acids. That might have been useful to know. The contribution of the 2,2,6 versus the others that were selected in ferrets.
That may be forthcoming.
Probably. So basically you see how this mutation Q226L, how this affects binding to human sialic acid, how it improved it, and how it decreased binding to the alpha-2,3 sialic acids. And it's very striking. The sialic acids fit in very differently in these pockets. You have to look at these pictures to really appreciate it. But they're almost flipped around in the way they're interacting. And again, the point here is that the transmissible H5 binds sialic acid in a way that human strains, known human pandemic strains, bind alpha-2,6 sialic acid. So I think what it says is that selection in the ferret has made this receptor interaction similar to a human virus-human receptor interaction. Okay, just that one part, which is not the whole thing about transmissibility. You need obviously other mutations as well. But what's been done here in the ferret is clearly relevant, at least mechanistically, for human infections.
And I know I say this repeatedly, but I am just constantly amazed at the speed at which some of this stuff comes out.
I was just going to say, it's been a year since these came out, and here we have structures.
We're looking at structures and binding constants and all this biophysical detail of individual amino acids, and the only complaint to come up is gee, maybe they could have mutated an addition, or looked at an additional mutant in the paper. It just it's jaw dropping how fast this is moving ahead.
It used to take years to get an atomic structure from a protein crystal, and now it's nothing. It's amazing.
Let me read just one sentence which sums this up nicely. They say the properties of the transmissible mutant described here suggest that H5 viruses could take a similar evolutionary pathway in humans to that followed in 57 and 68 by avian H2 and H3 viruses. And again, that's because that mutation, Q226L, is in the transmissible H5 and in those viruses as well, and that allows the transmissible H5 to interact with alpha-2,6 sialic acid in a way that human strains interact with it. So that's nice. It's a nice study.
Yeah.
The first structures took years. Sialic acid HA structures took years to do. Yeah. I remember John Scahill was involved in those ages ago. So this is cool.
Once again, important insights coming out of these papers that there was a lot of fuss about and discussion of redacting them.
And you couldn't publish this if it were not okay to publish the amino acids.
There you go.
Changes
You
in
bet.
those.
Sure.
This paper would be completely banned.
Yeah.
Kathy?
What I was going to say is relating back to what Rich said about ordinary people could do this kind of thing. I think I'm, and maybe you guys too, are affected by thinking that, X-ray crystallography and solving structures is, really hard, and it's something that we'd have to spend a lot of time learning. But I was talking to a crystallography friend of mine last weekend, and he said it's really quite routine now. And I said, so it's almost like kits? And he said, yeah, pretty much. So that's part of why, a lot of structures come out really fast, and from labs where you might not think that they have the structural expertise, but it's just another fast-moving field in science.
So isn't it still iffy to make a crystal in the first place, though?
Yeah, I think that's still magic.
Mm-hmm. But they have these robots and these trays and
Yeah,
all
that's
that
actually
kind of
not magic. It's brute force.
Yeah.
It's I've researched a couple of stories on that, and the technique is still just you don't know what's going to work, so you try everything. But the robotic technology and the automation of the process is so good now that you can try everything.
And sometimes it works and sometimes it doesn't.
Yeah, but you set up 1 million experiments and one of them works, and that's how it's done.
Anyway, I want to repeat what Alan said. If the original papers had not been published, the ferret transmission papers, this paper would not be able to be published. And this is full of really important stuff that everyone needs to see. So, I mean, there were some scientists who argued against publication as well. And I think that I understand where they're coming from, but this is great stuff, and this is pushing the field forward. I mean, I would say that our understanding of HA receptor interactions is huge now as a consequence of this. Almost better than most other viruses.
Yeah, and this gets us closer to real-world applications. I mean, this is fundamental information you're going to need if you ever want to get to a point of making reliable predictions about what could become a pandemic and what couldn't.
Right.
You need to know what are the ingredients that make something capable of jumping from one species to another, and here you go.
Yeah, remember our discussion last week about that? We were saying someday we'll be able to look at a sequence and say, this is a pandemic strain.
Right.
So someone put a comment on that TWiV episode, GSGS. He said, I have to find this here. He actually went through and indexed the episode, and all the places where we started talking about H7, he indexed in the comment section. He said, how in some decades we may understand how flu goes pandemic, and then he wrote as a comment, but do we want that? Dual use. It's a good point, right?
Yeah.
Because then you'll be able to say, here is a pandemic sequence. Go for it. All right, let's do some email.
Sure.
Okay, so we're skipping the third paper then. Okay.
We'll do that next time because I think we don't have enough time for it to justify it. Mm-hmm. And we will skip
that email that goes with it.
The email that goes with it. And that brings us to Cliff. And Rich, why don't you read Cliff's?
Okay. Cliff writes, hello. Regarding your ongoing discussion on vaccinations, here is my argument when the other person knows me well enough to know I am not serious. After exhausting the list of arguments in favor of vaccination, I like to respond, well, we have to limit the population somehow, and I'm vaccinated. Unfortunately, it is no more effective than any other argument since their response is usually a story about how they know a vaccinated person who still got the, quote, stomach flu, unquote. Apparently, I cannot convince them that the stomach flu is not influenza. Cliff. Yeah.
Yeah.
Yeah, I don't know. Population limitation is a bit severe.
Yeah.
But thanks anyway, Cliff. Kathy, can you take the next one?
Sure. Ashley writes, "Hello, professors. Just want you to know you have a huge undergraduate fan from China. I'm always very interested in virology and immunology and would like to pursue them in my future study. I remember you have talked about coronavirus the other day, and since it's been 10 years since SARS struck the world, especially China, I would love to know more details about this history. As far as I remember, the disease broke out during the Chinese Spring Festival in 2003, and the first case happened pretty near to my home city, Guangzhou. It was a tough time for everyone when I was a kid then, were told not to go out and talk to others, and I heard people dying in hospitals in the city. But sometime later, the virus just disappeared, and it seems like it never has come back. I've heard from my virology professor that scientists at the time were working hard on this virus, but it disappeared so that they had nothing to push them to keep on working. I'm wondering if there were specific measures taken at that time in addition to washing hands and avoiding going to crowded places, and if there's any chance the virus might come back, or is it gone like smallpox? Cheers. Ashley, and she's from Zhejiang University in Hangzhou, China.
I actually had an episode on SARS with Eric Donaldson where he sort of talked about the history. That's number 28. You might want to check that out. It was also, since it was the 10th anniversary, there was a great series of articles in Science very recently, and there's one by Ensrink which goes over the history, which is really good. I don't know if you'll be able to get that, Ashley, but I would recommend that one.
Yeah, it's behind a paywall, unfortunately. But
too bad.
Yeah.
Maybe through the university she can do that.
Yeah, or if your library, if you want to go old school, you could go down to the library and see if they have the paper issue.
So I just did a lecture this week on emerging infections, and part of that was SARS, and so I'm up on that. And it turns out that the next year after that, so the SARS thing was confined mainly by quarantine and travel restrictions and public health awareness because there were no vaccines and no antivirals. The next year, there was an outbreak of 4 people in the same city, Guangzhou, and it was a different strain. It was a separate introduction from an animal into people, and that strain had one less mutation in the receptor-binding site of the spike protein of the virus, and so was not able to bind as well and didn't spread. So that was really instructive in a way. But apparently these precursors are still circulating in wild animals in China. You know, they have banned the use of wild animals in the live meat markets, but you know that people probably get around that in various ways. There's a book by David Quammen called Spillover, and he talks about going to the meat markets in China and finding people who will sell him wild game. You know, so
yeah,
it's not perfect anyway. So it could come back, yeah.
Do we have any sense of how important the public health measures were in containing this infection, or whether it would have just burned out by itself?
I don't think anyone can answer that, right?
Yeah, it's kind of, you can't do the controls, right?
Yeah.
I mean, it's nice to think that the measures, like scanning people at airports for fever, right? But I don't know. Yeah, there's no controlled experiment.
It's nice to think that, but it's also kind of hard to believe.
Yeah, in a way, right?
Because the scanning people for fevers, you know, somebody comes onto your plane. Is anybody feeling ill? You'd have to be an idiot to say yes. Of course nobody's going to say they're feeling ill. You're going to get pulled off the plane and hauled off to a hospital. No, you want to go home.
Yeah, one of my students, virology students, we had office hours yesterday, and she said a friend of hers was leaving China, and she went through the temperature scanner. She had a temperature, and they quarantined her, you know, until your temperature's gone. So
yeah, I remember I was in China in 2006, and part of the departure procedure. How are you feeling? Fine.
But Rich,
it's amazing. This was
Go ahead.
Sorry. It's amazing. This was 10 years ago. That's all I was going to say. Time really flies.
I don't know, why would it just burn out? You know, if it were really well adapted and transmissible, which it should seem to be, I don't know.
Maybe it was seasonal. Maybe
it's gone.
But yeah, and it's gone. It's gone. I mean, part of it probably was the quarantine procedures, because it did have to get from person to person, and everybody became, I remember at that time, everybody became totally paranoid about illness, and everybody in China was wearing masks, and that probably had some impact on it.
But if you compare it to something like a flu pandemic, you know, you could do all that stuff, and you're not going to stop a flu pandemic.
Right.
Yeah,
boom.
Probably not, yeah. Because even if you can't travel, it'll just spread by foot, right?
Mm-hmm.
Yeah, well,
So-
H1N1. Go ahead, Kathy. Sorry.
No, I'm have a different topic, so go ahead.
No, that's
we're done.
Right.
We're done.
So, Ashley's last question, if there's any chance the virus might come back or is it gone like smallpox, relates to something that we're going to talk about probably next time in a nice story on dengue. But these emerging viruses, when they arise from an animal as a zoonosis, there is the potential for that to happen again. And
Sure.
like you just described, Vincent, the next year there was something and there were only 3 mutations, but it's conceivable that something like this could come back. So no, it's probably not gone like smallpox is gone.
Because smallpox infected only humans.
Right, and
No,
that coronavirus we were talking about a few months ago in the Middle East, we haven't heard from it lately.
Yeah,
Right.
Yeah.
There's another coronavirus outbreak that seemed like it might be going somewhere, and now, boom.
Right, which is good. That's fine. We don't.
Oh, yeah,
we're
we
not-
don't need to
We're
hear
not
from
complaining. Speaking of smallpox, there was quite a large article in Scientific American about the monkeypox stuff in Africa, Rich. I don't know if you saw that, but,
No, I did not.
as you know, because there's no more smallpox immunization. We talked about this. There's more
Right.
infections of people with monkeypox virus.
Right.
But unfortunately, the headline on the Scientific American article was, is this the next smallpox, which is really
Yeah.
bad.
We just had a visit here last week from Inger Damon from the CDC, and she went over
cool.
she went over all that stuff. It's pretty interesting. There are a lot of, well, we've talked about this before too. There are there are a lot of pox viruses that were covered by the smallpox vaccine
Yeah.
that now that we don't have the smallpox vaccine are popping up here and there.
They're annoyances, right? They're not
They're
really...
they're mostly nuisances. Yeah, cowpox is one of the most notable. That's showing up in Europe a lot, and it's a nuisance. It can be, a pretty big nuisance, but it's not a lethal disease.
Yeah.
Right. This is the concept that also is in this paper called the empty niche. So
Right.
where a virus eradication by vaccination provides an empty niche for some other virus related to then come to the forefront. It's a nice concept.
So in those outbreaks of cowpox, does Edward Jenner show up? Okay, so the title of the Scientific American article, which I complained about, I want just to be accurate here. Oh, nice. Continued to cite. Could monkeypox take over where smallpox left off?
Right.
All right, so that's
Just
a bit much, isn't it?
Yeah.
And they
Yes,
have a
that's
picture
a bit
of
much.
they have a picture of a kid with smallpox, and it makes people think that this is what monkeypox does. All right, I want to read the next one because it is from Lisa, who's out in Berkeley. Dear TWiV team, while listening to a recent episode, I heard a request for a short video to use in response to vaccine doubters. Here's my favorite one for flu. This is a funny video.
Yes.
I'm looking forward to meeting Dr. Racaniello at the Microbiology Student Group Symposium at UC Berkeley in a few weeks. Best, Lisa.
Yeah, it's I guess a Canadian comedian.
Yes.
Maybe that's redundant, but he has this nice little riff about the flu vaccine. It's good.
The only
Yes.
thing is, I hate the canned laughter that they stuck in.
Yes.
They didn't need that.
No, they didn't need that at all. That should go.
And I really like the graffiti. There's this huge wall of graffiti. He's walking down this street.
Yeah.
Really neat graffiti.
Mm-hmm.
And he says at the end, it's just a little prick. Don't be one, get one. That was good. That could be a title one day.
Yeah.
Mm-hmm.
Okay. Who is Alan, right? You're next.
I think it may be my turn.
Good.
Dave writes, how I came to be a TWiV listener. Science 360 Radio on TuneIn Internet Radio, remarkably loaded with content from down under and up north, leaving the U.S. looking stunted relatively. Purely serendipitous encounter as I don't systematically search. Worth recommending to others. You guys totally engage me in all 3 TWiVs. Much fun, thanks. Retired RN, partly trained medical anthropologist, fully trained child adult clinical psychologist, too scientifically minded to be content clinician, too poorly disciplined ADD to be professional academic scientist, to which I'd otherwise have aspired, irrepressibly curious about the natural way things work. Info flow about which is my salvation. Grad school time spent in lounge reading science and whatnot rather than tending to business. Find I need psychomotoric activation in order to function well and can't sit still for academic responsibilities making nursing viable. I have no regular productive activity, isolated. I have only dated bonehead education in micro detail, but find it easy to follow your conversations. Thanks, because I keep on learning, which is my primary value. Would do something to hang with and assist somebody if enticed, e.g. can critically edit written material. Now have forgotten how your discussion of your podcast inspiration prompted my little spate of applause and self-disclosure here, but glad you're doing your public bit.
This is great.
Dave in Fresno.
Very good.
Just great. I love it.
That's quite a bio.
Yes.
Mm-hmm.
How I came to be a TWiV listener. That's just great. Who's next? It must be Rich, right?
Must be Rich, yes.
Okay.
Oh, you got a long one there.
Mary Ann writes, "Hi, TWiV team. I've been listening to TWiV and now TWIM and TWIP for a while, and every episode I feel inspired to write in with a comment on the show. I'm finally getting around to it. I feel like I'm a virologist by association. I have a PhD in immunology, and I was fortunate to receive some amazing postdoctoral training in the lab of Grant McFadden just prior to his move to UF. I've had the pleasure of meeting Rich on several occasions, and so hearing him on the podcast is almost like chatting with an old golfing buddy. I went to work with Grant to study how poxviruses manipulate the host immune system, but quickly was steered into his then new project studying the oncolytic properties of myxoma virus. I then spent the next 7 years studying poxviruses and how they're able to infect and kill cancer cells during two very challenging and interesting postdoc positions. Clearly, on the academic track, I then took an alternative path, a position as a scientist in a small biotech here in Canada that focuses on vaccine development. In the past year or so that I've been listening to TWiV, I've often wanted to comment on topics such as postdoctoral training, alternative careers in science, the correct pronunciation of factor 10,
Good job.
10, not X. That had me grumbling all day, and oncolytic viruses. However, the one nearest and dearest to my heart is the ongoing challenges of vaccine development and its perception by the general public. In a way, I may have a unique perspective. Since taking on my current position, I've also welcomed my first child. I have spent a lot of time in the company of other new parents, and invariably, vaccines have been a frequent topic of conversation. It's amazing how fast all the myths surrounding vaccinations appear, and I feel compelled as a scientist armed with facts to debunk these myths. Let me say that it is an ongoing challenge, but one that is worth the effort. I try to provide parents, those who either ask for my opinion or those who impart clearly false information as fact to other parents, with unbiased information from reputable peer-reviewed sources. Needless to say, I've been accused of pushing the pharma agenda a couple of times. However, many parents, especially the parents of children with chronic conditions who are at high risk of contracting serious diseases, appreciate someone who is passionate about the advantage of vaccines. I commonly recommend the panic virus as a reference to anyone who wants to learn more. And as anecdote is a surprisingly strong tool, I commonly tell people that I understand all the risks and benefits of vaccines intimately, and my own child is fully vaccinated on schedule. I think this all harkens back to a very good point brought up in the last TWiV regarding the certainty in which anti-vaccine advocates speak about the dangers of vaccination. It is a very clever ploy because they understand that this is exactly counter to how scientists speak about science. They precipitate this perception among the non-science public is that this careful way scientists describe findings is because either they don't know the correct answer or they're hiding something. This facilitates the disconnect with the public, who may believe that science is a collection of facts rather than a collection of findings that require careful and thoughtful interpretations that may change based on the next set of findings. A part of the solution to this rests squarely on the shoulders of the scientists themselves. Science outreach is so important, and I know I'm preaching to the choir on this one, as podcasts such as this one do so much to make science accessible to all. However, the importance of being a good science communicator, especially the talent of being able to communicate your science to a non-specialist audience, is rarely taught and not nearly as valued as it should be. Here in Canada, as I am sure there are in many other countries, there are wonderful outreach organizations. The one I'm most familiar with is Let's Talk Science, and she gives a link. An organization I've been involved with since I was a graduate student. These programs teach graduate students to be comfortable with science as they share their knowledge and enthusiasm to school-aged children. As I've said many times, much to the chagrin of colleagues, that if you can't explain your research to a 10-year-old, you don't understand it well enough. Here. Mm-hmm. The idea of stepping away from terminology and jargon and getting to the fundamental questions isn't easy, and it takes practice, but it is so important. As our science advances and becomes increasingly technical and abstract, it's easy to lose sight of the fact that much of that science is paid for by public funds, and they need to understand its importance. Controversies around vaccination, a practice that has been saving countless lives since the time of Jenner, is only the tip of the iceberg. On the topic of science literacy, I do have a pick for you. The Cold Spring Harbor Lab Press has a series of 4 children's books known as Enjoying Your Cells series. It includes Enjoying Your Cells, Have a Nice DNA, Gene Machines, and Germ Zappers. I received Germ Zappers when my son was born, and although the text is a little over his head at 20 months old, my 6-year-old niece loves the book, and it is required reading every night she's visiting. She has learned a lot about cells and the immune system, what sort of germs exist, and the value of vaccination, including a special mention of vaccination against polio. Science literacy starts early. A link to these books can be found here. She gives the link. Keep up all the great work, and I'll be listening every week all the way to the end, just in case there's a small Easter egg thrown in. Cheers to all, Marianne. What a nice letter with a nice pick.
Nice, very good.
And here, yes, communicating science to the public is absolutely essential. It's a responsibility.
A lot of good points. Yes, very nice, Marianne. Thank you. I know it's hard to write all these things, but thank you for doing it because a lot of people will love it. Yep. Good stuff. All right, let us do some picks of the week. Rich Condit, what do we have this week?
I have a pick that came to me from a graduate student, an ex-graduate student, Candace Bishel, via Nicene. Mm-hmm. And it is a time lapse of videos of the sun, time lapse of pictures of the sun over a 3-year period, collapsed into 3 minutes. And basically, you see a lot of sunspots and other activity rotating across the sun. And there's a couple of little other things that flash by, like an eclipse and Venus going across the face of the sun. And it's really pretty cool.
Yeah.
Gorgeous.
The link I picked is from, I looked around for the original. It's a NASA video, but the link I picked was from NPR because it's nicely annotated, and it's got this composite picture at the end, and it's very, very, very pretty. And actually, what's even cooler about the NPR site is the comments. One is, disco balls all over the world are feeling very inadequate now. And another one, I really love this, because they at the end they show that this is only at one part of the spectrum that they've used for most of this, and then they show what other spectra look like. And somebody comments, this video clearly shows NPR's journalistic bias against the visual spectrum. NPR claims to be balanced, but most of this video shows only the sun at 171 angstroms. This just proves that NPR is another ultraviolet mouthpiece.
Great. That's great. Nice.
Anyway, it's pretty.
Good.
Thank you.
Excellent.
Alan, what do you have?
I have something that's not for another month, but it's probably a good idea to let people know about it now because I think they're probably selling out. In fact, they've sold out a couple of their things already. The World Science Festival, which is held in New York. I don't think I'll be able to make it, but this looks like a heck of an event if you're anywhere near town. It'll be May 29th to June 2nd. And they have lectures and events and all sorts of stuff. Some movies, some a science street fair, just a tremendous science outreach thing aimed at the general public.
Outstanding.
And it's at really venues cool. Around town. You can some of the things require tickets. The talks, they have limited size auditoriums, but others like the Science Street Fair, you can just walk into. As I say, I probably won't be in the area for it, but if you are, I would highly recommend putting this on your calendar.
How long has this been going on?
Since 2008.
Okay.
Hey, Alan, how do you get to do something in this? Do you know?
You should contact them.
I think you should come down one year and we could do a TWiV.
Do a TWiV. That would be great.
Don't you think they'd like that?
Yeah, absolutely. Probably best if you get in touch with them. There's a contact. They've got email and phone number, and you can.
Yeah, we could get some virologists from New York and.
Absolutely, that would be cool. Awesome.
It'd be a lot of
This
fun.
is outstanding. I just want to go.
Yeah.
It's great.
Wander around and see all these cool things.
Yeah, it's really matured into a very, very impressive thing.
Nice.
Excellent.
Kathy, what do you have?
Well, I have something that I got almost a week ago, and then it turned up this week on the astronomy picture of the day, and I wrote to you guys and said, I've been scooped, because I figured that from previous pick, everybody's already looking at the astronomy picture of the day. But this is a Canadian guy who's the head of the space station on the International Space Station right now, as I understand it. And he's taken questions from children. What happens if you do this in space? How do you do this in space? And a couple of girls sent in, what happens if you wring out a wet washcloth in space? And so he sets it up, and I won't tell you how it turns out. It didn't turn out how I thought it would, but it's just fascinating to see what happens. And then, again, because like Rich, I looked around for the best link. I chose this one because from that, it leads you to the other YouTubes of the children asking questions at how you do things in space. How do you wash your hands, make a peanut butter sandwich? How do you trim your nails? How do you shave? Various things, which just were really kind of interesting to watch. And he's just got a very nice, natural way of explaining things that I really enjoyed.
Yeah, this guy is really, really good. He also he tweets and sends out amazing pictures from the space station. It's really cool stuff.
So
he's got a Facebook presence that I follow, and there's a bunch of pictures of the planet every day with his comments and other things. He's very he's obviously passionate about communicating this stuff to the general public, and he does a great job of it. Chris Hadfield.
Yes.
Okay.
Wow,
Hey, Kathy, this isn't the same guy who did the space station tour that you picked a while ago, is it?
No, that was it was a woman who did that. It was just before she was going to leave. It was an American woman.
That's right. Oh, this guy's great.
Look at this, the interior. Look at all those wires.
Yeah.
I just scrubbed to the end, and you can picture him holding a mic and talking, and this thing is full of wires. If you get break, what if you hit one of them when you're spinning around? Man, it's dangerous.
For a lot of the video, he doesn't actually hold the mic.
Right.
Yeah. It's
It
just
just floats
floating.
in front.
It's floating in front of him. Look at that. Yeah, that's great. Nice. It doesn't go anywhere, right? So
Right.
this thing is still up there. Is that right? In space?
Yeah. Oh, the space station.
And how do you
Or the
get to it?
washcloth.
How do you get to it? No.
Nowadays, you get to it in one of the Russian
Soyuz thingies.
modules.
Yeah.
Yeah, one of the.
Wow.
Yep.
So we have to pay the Russians for that, huh?
Yeah, they need the money.
I thought they had a lot of oil.
They do, but I don't think their space program is terribly well funded these days outside of.
My pick is a repick. This was a book picked by a listener some time ago, but I wanted to repick it because it is just awesome. and it's called The Origin of AIDS by Jacques Pepin. And I just read it a few weeks ago.
Yeah.
And this book is written beautifully. So this is a Canadian physician who has worked in Africa, and he's also published a variety of papers, which you can find in the PLOS journals. And he traces the story of the emergence from, the first isolation of viruses, the phylogenetic information that led to the idea that the main group of HIV-1, the M group, originated in the early 1900s from a chimpanzee infection of a hunter and so forth. And then he goes through a lot of evidence that the amplification of the infection was a consequence of the colonization of Africa by Europe, the introduction of prostitution, and the health care imposed by the Europeans, which unfortunately utilized needles that were used over and over again and helped spread various diseases. And it is just riveting. So you have to read this. It is great.
Okay.
He writes beautifully,
Yeah.
and the science is awesome. He explains, he has the first explanation of a phylogenetic tree that I really understood. So
I could use that.
He's really good. So I totally recommend this, and you will be amazed. And it's basically a chimp, a hunter, 60 million infections. Amazing. Just amazing. So go read this.
Okay.
Origin of AIDS. You will not, and if you don't like it, I will give you your money back. Because you're going to love it. This is just so cool. We have a listener pick from Mark, who writes, "Hello, Team TWiV. I have been listening for about 4 years. TWiV is the only podcast I listen to 100% each week. Keep up the good work." Episode 229's opening with the story of Hillary Koprowski preparing a mouse brain cocktail in a blender to test his polio cure by drinking it was a riveting tale. This is the second time that I recall a blender having a role in the episode. The other, if memory serves, was on sister podcast TWiM in the episode at Cold Spring Harbor Lab, an historical look at equipment used there by XXX, whose name memory fails to record. That would be Al Hershey and Martha Chase. Are you familiar with Blendtec, makers of industrial blenders? They use internet video and viral marketing campaigns. In each video, Blendtec conducts a simple experiment that asks the question, will it blend? Where it is an object ranging from an iPhone, all models, and the original is tested here, to an avocado or Halo 4 video game DVDs. The it is put into a blender which is powered on to conduct the experiment. Viewers are warned, do not try this at home. The results provide insights about products and materials, but they mostly provide laughs. Science has no boundaries, so there is even a Will It Blend iPhone, iPod app available, so the scientist on the go can consult the latest in Will It Blend literature. Cheers. Thank you, Mark. That's very cool.
These are a riot. I've played the iPhone blend now to probably a half a dozen people, and I even have the Will It Blend iPhone app on my phone now.
You do? Yeah, I'll have to get that. That's really cool.
I remember seeing the Will It Blend iPhone app shortly after I got my iPhone, which I was guarding very carefully because I spent a fair amount of money on it, and I just thought anything could happen. And then I thought, I just couldn't believe that they would put a phone in a blender and ruin it.
Yeah.
Yeah.
It's too bad.
I particularly like the golf ball one. Anybody who's who's a golfer had a tough day on the course will enjoy seeing him blend up a bunch of golf balls.
They're full of wound up elastic bands, right?
Yeah, not for. Actually, nowadays they're filled with some kind of goo. It's the elastic band version, I think, is gone. They have goo and more solid core. But that used to be the case. You must have taken a golf ball apart when you were a kid.
Yeah, it was all rubber bands.
Yeah, it was all rubber bands.
Not anymore, huh?
Not anymore, I don't think so.
I still have a scar from where I tried to cut open a Super Ball, though.
And that is TWiV 230. We do this every week on a Friday usually, and you can hear the released episode on Sundays, which you can find at iTunes at twiv.tv. And if you like what we do, go on over to iTunes, subscribe, and leave a comment or rating to keep us visible there on the directory so more people will find us. And send your questions and comments to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This is a lot of fun, and I learned a lot.
I will see you next month in Denver.
That's correct.
We will do a TWiV together. And just to keep the University of Michigan busy, I'm doing a TWiM there, and I'm going to have your colleague, Michelle Swanson, on that.
Excellent.
You know her,
That'll be
right?
Good. I do. She's just down the hall.
Yeah, it'd be cool. Keeping you guys busy. Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
My pleasure. Always a great time.
And you are healing your shoulder up, right? You're all good?
Yeah, I just came back from the doctor this morning. I'm in good shape.
No more sling?
I actually, no more. I got rid of the sling about a week ago, and I now have the mouse on the right-hand side of my computer. And, things are coming. It's amazing, I mean, when this is all done, the collarbone's going to have its original shape. These little cells go in there, and they're just polishing and remodeling and making it all right. It's incredible. So I'm doing well. And I'm out of here for 2 weeks.
Oh, yeah? Where you going?
Italy.
Nice.
Cool.
Good. Yeah.
Nice country.
Yeah, it'll be
All
good.
right, have fun.
I will.
Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
You're not going to Italy, are you?
No, so unfortunately not.
I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.